It's, it's really great to see everybody here. The UK is absolutely amazing at the life sciences, and you all know about the Nobel Prize for Crick and Watson, but we've probably per capita had more Nobel Prizes in the life sciences than any other country. And because of this really vibrant science base, it means we've grown really world-class pharmaceutical companies and biomedical companies that not only make us live longer, live better lives, also return a massive amount of investment to the UK, one of the biggest contributions to our GDP, and employ hundreds of thousands of people. So it's very, very important. And the work of me and my group is about measuring the chemistry of life. And I work a lot with the pharmaceutical industry, and I'll tell you a little bit more about that, in that, I don't know if you know, but it takes about 10 years to go from the original sort of idea of a particular drug molecule, this is a typical kind of drug molecule, from those early days of knowing it has some biological effect, right through to a medicine that you can take, takes around 10 years. And it takes billions of pounds of investment to do that. So there's a huge amount of work that goes on, and I'll talk a little bit about more, to try and optimize all of that so we can get the drugs sooner and also make them more efficacious, so we can make them more effective to people. And also, I work a lot with the leading biomedical researchers, so we're very lucky in the UK, as I said, to have a very strong academic base. I particularly work a lot with the Francis Crick Institute, which is one of the biggest biomedical research institutes in the world. And so a lot of my research is around the cell. So I don't know how many people are kind of familiar with the cells. I'm a physicist by background, but over the last 10 years or so, I've moved more and more into biology. And I still amazed by the cell. So here is a cell. I can pass it around. I don't know if anyone wants to have a look around and move those around. So that's about 5,000 times the scale of a normal human or animal cell. And in the center there is a nucleus, as you'll know, where the DNA is. That's the information that codifies basically everything that then goes on with our bodies. So this is your DNA and the genetics. They then do something called the RNA, which is the transcriptome. So those are the kinds, they send the messages to the other parts of the cell, which then make the proteins. And the proteins are then the things, the machinery within the cell that gets things done. It makes the different parts of the cell. It does the different things in the body. So that's something called proteomics. And then the proteins and everything that's going on in the cell is creating information, really. It's essentially a form of information. This is the chemistry of life. It's the metabolism. So you will all sort of kind of know, some people have a higher metabolic rate than others. So that's the way you process the chemistry that's going on in your cell. But it's amazingly complicated. So our body is, of course, mostly made of water, right? And the cell is mostly made of water. So the first job it's got to do is contain the stuff inside the cell and stop it just going blur everywhere. And it does that very cleverly, just like you see when you're doing the washing up, if you have a bit of oil that goes onto on the nice in your nice sink in the water, you'll feel it beads up, of course, because it's hydrophobic and the water's hydrophilic. So the cell is made out of lipid membrane. These are molecules which are very much like a fat. It's a long chain of carbon atoms, but they have a little polar head group in. So the polar head group part is happy to be in the water, and the long chain part doesn't like to be in the water at all, so it sticks out. And they automatically, if you drop these kinds of lipids into water, they immediately assemble a sphere. It's very, very clever. It's incredible, really. And if you go down to the engineering biology lab in Module 8, you'll see the amazing work that Max Riadnoff and his team are doing, engineering these kinds of structures from first principles. So we have this membrane. If as we talked earlier, if we want to get a drug into that cell, not only do we need to deliver it to the right cell in the body, but we've got to get it through that membrane. And that membrane is, as you know now, is hydrophobic. It wants to expel things that are water-like. So this is a typical kind of drug that we might want to look at. This is a drug called amiodarone. It's a drug used in arrhythmia. If we want to get the drugs into the body, we normally take it through a medicine, a tablet, and then the drug has got to be soluble in some way. If it's not soluble, then it'll be like dust, and we won't be able to get it actually into the body and around to the affected part of the body. So that means it's got to be kind of polar. It's got to be happy to be in the water. So once the drug then gets towards our cell membrane, it then sees this layer here, which is hydrophobic. This molecule's hydrophilic, and it won't go in. So a lot of work goes on in the pharmaceutical industry to try and tailor the properties of this molecule. We can see these red things are oxygen atoms, so they're going to make it kind of more hydrophilic. These are iodine atoms. The black things here are carbon. The loss of carbon in there usually makes it hydrophobic. So a big challenge for the pharmaceutical company is to design the drug so it has the right biological effect when it gets to, usually these things interact with a protein molecule, and that protein molecule then is disrupted in some way, and that then is used to treat the disease. But if we don't get it into the cell, that's the problem. So if you see, for example, a good example of this is, for example, the messenger RNA vaccine that was developed by Moderna that we all knew from the COVID times. The RNA is a very polar molecule, so that won't go into the cell at all. But it's actually delivered in a lipid vesicle. The lipid vesicle surrounds it with something which is really hydrophobic, and then that will come towards the cell membrane, and then it will go in towards the cell. So this is really starts to be a lot of very fascinating processes that go on. So I just wanted to start off with, so this is a quote from a Nobel Prize winner, Sidney Brenner, who I find really inspiring. And what he said is, what we really need to learn from this is that mathematics is the art of the pure, physics is the art of the optimal, and biology is the art of whatever wins will survive. And this is exactly what I found. Biology is so complicated because of evolution. So what do we really mean by life? So one of the sort of tests for life is, can it evolve? And can it sustain itself in its own environment? So yeah, the other reason I really like this talk, this quote, is because it brings together kind of the key things that we need as scientists to be able to make progress in this field. So in my group, we bring together people in mathematics, people in physics, people in biology, to be able to look inside the cell. So everybody's got the cell now. If you see inside that now, let's say we've managed to get our drug molecule into that cell. It's only a few microns in size. And if I'm now trying, as a pharmaceutical company, I want to know, have I got enough of the drug into that cell? Where has it ended up in that cell? That's where measurement becomes really key, because if they can have that information, they can then see if that drug is going to be effective. And that's exactly what we do here at MPL. And the other things that we need to take care of is, remember, we have this 10-year timeline. What if that molecule is actually going to the wrong cells and causing a problem? So this molecule here, amiodarone, is actually well known. Although it's a very effective drug, it does go into macrophage cells, and that starts to have a side effect. And then we can, using the techniques that my group here at MPL are developing, we can see into those cells and see what's happening. And that's really important information for the companies developing the drugs, because they can then tailor these designs to be able to optimize where the drug goes to particular cell types using better delivery mechanisms. The other thing is that inside the cell, there's lots of different compartments called organelles. And if the drug goes into one of those organelles, this could also be a big problem. So the mitochondria, as you probably heard, that's about a micron size, looks like a bacteria inside the cell, like a sausage shape. And that's the energy factory of the cell. If drugs get in there, that can really be a very big problem and can cause the cell then to die. And obviously you don't want that. So looking for very early signs of the drug going to the wrong place can then save 8 years of research that in the end would be wasted. Likewise, there's the lysosome is another part of the cell. This is where this drug often can end up in. So if we can spot all these things earlier, we can help make better drugs. But it's actually very difficult to C, can you imagine the chemistry in the cell? There are thousands and thousands of different molecules. And we need to be able to sort of look into that cell, which is a tiny amount of liquid, and identify what exactly all those molecules are, where they are, and how they interact with each other. And that's exactly what we do at MPL. And if you, my lab is currently open in Module 8. And if you go over there, you can see the imaging technology that we invented here at MPL. And it's now in use all the way around the world. This it's actually really the most powerful molecular imaging technique that you can get. And we are using it also to sort of look at fundamental biology. So I think this is just how this kind of thing works. So we normally take something like a tissue sample here, or it could just be a single cell that we can do that. So we use, in our case, we use something called an ion beam, which is a very focused beam of ions. It hits the sample surface. Every time it hits the sample surface, it liberates molecules that go into the gas phase. So these molecules could be the drug molecules that are in the tissue. It could be the lipids from this lipid membrane. It could be the protein fragments, or it could be the metabolites, which tell us about how healthy the cell is, or this information changes that are going on. All of that then goes into something called a mass spectrometer, which basically just separates out all of this material coming off by their mass to charge ratio. And then once that's in the computer, we can select particular peaks, and from that we can then reconstruct an image. And the amazing things that we do at MPL with this is that we're pushing the limits of the resolution. So it's a bit like, you've heard of like the Heisenberg uncertainty principle, where you either know something's momentum really well, or you know its position really well. In biology, we have something very similar. So we can either know its identity really well, but then we have no idea where it is. So to do that, we have to use big techniques like nuclear magnetic resonance. Or we can use techniques which allow us to go to very high resolution. You might have heard of electron microscopy, but then we've got no idea what it is anymore. So it becomes a real problem. So in my group, what we've done is we've really pushed the boundaries of being able to know what it is and where it is simultaneously. And that's really been a big breakthrough, not only in biology actually, but generally in the life sciences and in advanced manufacturing. Yeah, and at MPL, we have the National Centre of Excellence in Mass Spectrometry Imaging, which is one of the world's largest centres. We have a whole range of these different imaging capabilities. And one of the methods, you can actually, it's used in surgery. So as the surgeon is cutting through, for example, some cancer tissue, it gets a readout into the mass spectrometer, looking at the metabolism of the cell. So the computer can then give a readout to the surgeon saying, yes, this is healthy tissue, or no, this is diseased tissue. These kinds of decisions for a surgeon are very powerful and helpful, because in the case of removing a tumor, you want to save as much of the healthy tissue as you possibly can, but you want to cut, and you want to make sure you cut out all of the tumor tissue. So by having this, and by doing the kinds of metrology that we do in MPL, it gives much more confidence to people taking those measurements. So this slide here is just really telling us about the things I did earlier in terms of seeing where the drug goes. I just wanted to show this slide because I'm a physicist, and my movement into biology was really encouraged by this very famous clinical pharmacologist, Sir Colin Dollery. He founded the field of clinical pharmacology. He was at GlaxoSmithKline. So his work, and by focusing on that, has been able to make drugs much more effective for humans, and been able to deliver them to patients much quicker. This is this instrument. It's so-called Orbisims. So this was the first one that I invented and was delivered to MPL in 2016. There are now over 20 of them around the world, and all of those are providing amazing answers to really tough biological questions that you can't get elsewhere. So this is the kinds of things that we're able to do. So if you see the cell there, we're able to actually track how much of the drug molecule is in those individual cells. We can measure this with really high accuracy. But the other thing that's really amazing about cells, so if I had genetically identical cells all grown together and all incubated in a bath with this drug molecule in, you would expect that they would all take up the same amount of that drug. But this is exactly where biology is so much more complicated than physics, going back to my earlier statement. And oftentimes in biology now, we're really trying to do things at the single cell level, because if we measure the whole population, it kind of masks out the detailed information that we need to understand. So now by actually measuring this at a single cell by single cell level, we can see exactly how much drug goes in the cell, and we can see the metabolic changes that occur. We don't need to really see the detail of this graph particularly, but what you can see is that some cells take up a huge amount of drug. This is the amount of drug. And some cells take up hardly any drug, but they're all following on the same kind of line. So this is really telling you the biology is really complicated, but through the physics and the methods that we're using, we can see that the principles are basically very similar. So this was really actually kind of revolutionary data at the time in 2017. And this is now providing really important information for people in biology trying to look at things at the single cell level. So I think that pretty much takes me to time, and thank you very much